Good morning and good afternoon. My name is Matt Cheeseman. I am the Digital Assistant here at European Pharmaceutical Review. Welcome to the latest instalment of our webinar series, today in partnership with BMW Tech. Today we'll be looking at the advantages of handheld Raman in the pharmaceutical industry. This webcast will discuss the benefits and applications of handheld Raman, including the reasons the industry is interested in the technology, data integrity, method development, how samples are scanned, and challenges that have been conquered. We'll also discuss PICS and how it is unifying the regulatory perspective globally, currently including 43 member countries. We'll discuss how these regulations are moving towards 100% container testing globally and how handheld Raman technology can help to achieve it, as well as the importance of validation of instrumentation and methodologies. We have two superb speakers lined up for you today to share with you their thoughts and expertise. Now, if you do have any questions throughout the webinar, you can ask these using the questions button on the left of your screen, and we will endeavour to answer all of these at the end of today's session. So without any further ado, please allow me to introduce our first speaker, who is Sulaf Assi. Sulaf is an Associate Lecturer in Forensic Sciences at Bournemouth University here in the United Kingdom. Sulaf will be discussing handheld Raman spectroscopy for identification of online drug products. Now it's over to you, Sulaf.
Okay, thank you, Matthew. Thanks everyone for attending today. And today I will talk to you about handheld Raman spectroscopy for identification of online drug products. To introduce the problem, the aim of this work is to identify and authenticate drug products purchased from the Internet. And these include two types of products, novel psychoactive substances and conventional medicines products. The specific objectives I'm going to go through in this work is first to construct methods and libraries of substances and products using handheld Raman spectroscopy. Second, to identify the novel psychoactive substances products purchased online. And third, to authenticate the medicines purchased online, whether they're authentic or counterfeit. So to start first, I will compare the novel psychoactive substances and counterfeit medicines. What's the difference? Both these types of products represent a major public health threat. The difference is that novel psychoactive substances are often unknown source and origin. So you buy the drug from the Internet, it is most of the time like a powder. Sometimes it's an attractive packaging. Most of the time there's not enough information on the drug. What is it? And it's often labeled either a research chemical or not for human consumption. The chemical purity is too much variable for this product, so you might have 100% purity, you might have 90%, you might have 10% purity, or even something else. For counterfeit medicines, they're most of the time labeled as medicine by a pharmaceutical company manufactured, and the MA holder is written, but in the real life it's a counterfeit. But you can find them in legal and illegal online pharmacies. They can be labeled correctly sometimes, and they can be of any pharmacological class. So you might have a medicine from lifestyle or life-threatening classes. It doesn't matter. The chemical purity here as well, it is variable. So when we are looking into a medicine, we're looking for something that is has therapeutic indication. For example, antibiotic, antihypertensive, anti-angina, something that is treating a disease. And the dosage form here, we can have variable dosage forms. So it can be solid, semi-solid, liquid. The pharmacological class, as I said before, it can be assigned, and there's always the manufacturer, where it was done. Is it branded or generic, which manufacturer, and the MA holder, who has the license for the product, and the source it's from. Normally, I can buy it from hospitals, from pharmacies, through the supply chains, and online, which we'll be talking about today, where we ordered the samples from. On the other hand, when we have a novel psychoactive substance product, it's intended for abuse. It has a psychoactive effect, and this is why it's taken. Most of the time, the samples I have are solid form, which can be tablet, capsule, or powder. Very few samples are liquid. Semi-solids, not yet. We have. The class and manufacturer, the pharmacological class, sometimes it says stimulant or psychedelic, but it's inaccurate. Sometimes, most of the time, it's contradicting what's inside the label. The label claim is not correct most of the time, and the additives is not known. The availability of this, it can be in head shops, parties, festivals, but it's mainly online in illegal websites. So this displays two pictures. The one on the left is a legal high or novel psychoactive substance, and it's white powder. It can be anything inside. So my aim is to identify what's inside it. The other one, we have a set of authentic and counterfeit tablets, Viagra tablets. I want to know which is authentic, which is counterfeit. So the aim to look at what is inside this tablet. So for tablets or medicines, so for these medicines, I would look into three options. Either it's authentic, so it matches the reference standard exactly with the physical and chemical characteristics. The other option, I would have a counterfeit, which is a medicine that is mislabeled. The identity is wrong, the source is not wrong, or it can be the label claim is wrong, or it has a defect in physical chemical properties. There is one thing to clarify is when I have a substandard medicine. Substandard medicines, they do not fulfill these specifications. If this is accidental, then they are substandard and the defect is in the manufacturing process. But if a substandard is on purposely done and deliberately done, then it is a counterfeit product and qualified as a counterfeit. On the other hand, what I'm looking at in an MPS product is totally different. I'm looking what is the pharmacologically active substance present in the product. Is there really a stimulant or a depressant or a hallucinogen? What is it? The other thing, what's the modalities of the intake and how is this affecting the action in the body? Is this dose dependent? Because most of the time I'm having new molecules. So is this a dose dependent thing or no? Is there any drug-drug interaction, drug-food interaction, drug-alcohol interaction? Additionally, what are these impurities, how they're affecting? And so there's a lot more questions to answer in this case, and they are critical. Both problems are increasing, and this slide shows us how there is a global crackdown. There was in June 2013 on fakes online, bought online fake drugs of $41 million, and this expands the problem, how is the counterfeits. As well, with novel psychoactive substances, the global rise is increasing, and according to the United Nations Office of Drug and Crime, we can see that there is constant theft of legal highs. And this makes our scenario with online drug products really critical, and stimulates the need for really rapid methods to identify these drugs and tackle them. And this is how the advantage of handheld Raman spectroscopy is that it's rapid, quick, easy to use, and with the help of online libraries, it can fit to both purposes, and this is what we'll see. So the instrument we have used here is the BWTech NanoRam, and this is the instrument, and it scans the tablet within seconds to less than a minute. And it can instantly give us a match, yes or no answer. So it can be used not only in the lab, it can be carried out to the field. If there is a market anywhere, straight away it will measure. It operates with laser excitation 785 nanometer, and the detector is charge-coupled device detector. The samples that have been used in this work, we've used reference products of substances and impurities present in the medicines and in the legal high MPS products. And for test products, I've used 7 internet samples. These samples were analyzed by reference analysis, and they are counterfeit samples when we done the reference lab analysis. So this is to check the potential of the instrument, how fast it is in identifying these counterfeits. The other type of products, we've used 6 MPS or legal high products that were purchased from the internet, and we have of unknown composition. I wanted to check them with this instrument. So this is details of the medicines that were used. These are the reference medicines for each of these, but each of these products was ordered online. So what we can see is a diversity of medicines. We have citalopram, doxazosine, Propecia, Reductil, Tamiflu, Viagra, and sertraline. We can see that citalopram and sertraline are for the lifestyle medicines for depression. We have Tamiflu, life-threatening for influenza, and variable. The doses of these are variable, so some of them are very low doses. For doxazosine and Propecia, for example, they're 1 and 4 milligrams. And this, we can see that there is a high percentage, the percentage of active ingredient will be low, and this is critical sometimes in identification. Whereas some have very high doses, like Viagra is 100 milligrams, sertraline 50 milligrams. The list of excipients in these products range from roughly, we have minimum of 6 excipients for doxazosine, and we have up to 20 excipients as in other ones. Most of them have microcrystalline cellulose as an excipient. And this, later we can see it affects the Raman signal. So these are the reference products, and we ordered similar products under the same label claim from the internet. And here we can see a little bit of background from the cellulose in the spectra, but we can see peaks corresponding to the authentic. The first one is citalopram, and the second spectrum below is the sertraline. And the background, it did not affect the accuracy of identification. It was characteristic for each product because each has a set of different excipients. The instrument inbuilt algorithm was used in this work for identification, and 2 modes were used. The first one is based on correlation, hit quality index, the one on the left explained in the equation, which compared the correlation coefficient between this reference product, the sample, and the sampled product. The other method is the probability-based method, and it's displayed on the right-hand side, and we can see it compares the probability between as well the reference and test products. So to start with the results, actually it first showed that it was a very rapid system. It was non-destructive. We scanned all the samples as they are and were able to carry out. The battery was had a long life, and the temperature, it was not affected by the temperature. It was selective as well to the speeches and the samples, as we will see going forward. So first, the library-based method, which is based on correlation. What was done with the medicine products, a library was creating, having signature of the 7 products that we saw. And this is the way, so it was very simple and straightforward. The first picture shows us the library, so straight away we can create a new library and then add samples to it, and the instrument has the option to scan the barcode of the sample. So that is really easy in identifying the products. So the first one shows us the Propecia, and the spectra is displayed below, and then we have the sample list. We did this for all the 7 product signatures, and the method was named Internet Medicines because it was used to authenticate the Internet Medicines. The probability-based operates in a different way. So a method was created for each separate product, and it had 20 signatures for each product, so 20 high-quality spectra. And this screen shows us how a Plavix method was created. The second screen shows the spectra, and once it's completed, it will say operation complete. And a separate method was created for each of the 7 products. So this way we have a library that contains, for correlation method, that contains all the 7 products, and a method for each separate product to compare the probability method. So in order to authenticate the things based on probability, what we did, there is 2 options. Either we authenticate the batch as a whole, or in case single individual tablets were purchased, because this is critical. When ordering a medicine online, you might get a product with a label, with a package and the full product, or you might get tablets in a sachet, and it's variable. It depends what you get. So the instrument has both options. So if we go to the batch mode, we can write the name of this, scan the barcode so it's stored, and the batch number, write it, the expiry date, all these can be completed, and then the scan will compare, and either you get, if it's matched, we can search. In this case, we have the Viagra tablet matching, and we can see both spectra, very high similarity. If it's no match, it will be displayed on the screen that is no match in the library. So first, before authenticating testing the products from the internet, what we have done is check the accuracy of the library method. So different tablets from the same batches, the authentic batches, were checked with the hit quality index, and we can see that the minimum, this is the R values, the hit quality index values based on correlation and how similar they are. And 3 values were compared for each product, and the mean value is displayed, and the standard deviation between the individual replicates measured are displayed in the table. And what we can see that only for doxazosine, Propecia, and Reductil, it's above roughly 84%, while the others are above 95%. And this is acceptable, but it's only for one reason, that these 3 products have relatively low percentage mass, and the dosage of them is 1 milligram, 4 milligram, and 15 milligram. So they have a high amount, percentage mass per mass of excipients. This is what made standard deviation between replicates a little bit higher than the previous ones. On the other hand, when we did the probability for these ones, we can find very high probabilities, the mean probabilities for all the products. And the threshold here is 0.05. We have far beyond, and the standard deviations are very low. And this was because the probability ignored the noise between this, while correlation is more of a linear method, and the noise interferes in this as well as the background. After that, we measured the counterfeit products, the internet samples that have been bought from purchased and analyzed. So, as I said, reference analysis showed that all these samples were counterfeit. And what we can see that here, the first column shows us the HQI values and the result, and the second and third and fourth column shows us the probability values and the results. So HQI gave no match for citalopram, doxazosine, and Reductil, and this is except because they were counterfeit. Apart from the others, only Tamiflu had 95% match, which is above the threshold. But this could be attributed to that the counterfeit, there was a little bit of similarity in the excipients in them, and this is what gave the whole signature interfered. On the other hand, probability algorithm we had 4 that gave pass, and they should fail: the Viagra, Tamiflu, sertraline, and doxazosine. So correlation was more accurate. The hit quality index was more accurate in identifying these products out of 7, the 4 products. So this is for counterfeit. Now to move to novel psychoactive substances products. As we said before, novel psychoactive substances or legal highs, you do not know what's inside the product. So we bought some products that were labeled as aminoindane and other products, and to see what's inside them. This is first a pure 2-aminoindane measured in the first upper picture, figure number 4, and we can see the signature corresponding to it. The second is caffeine. There are the 2 peaks for caffeine just before 1500 centimeters to the -1, and these were significant. Later we'll highlight them afterwards. And figure number 6 shows the lidocaine hydrochloride measured by the NanoRaman. So the 6 MPS products, most of them showed the presence of caffeine, and this is figures 7 and 8 shows 2 of these products. And we can see particularly in figure 8, the peaks around 1500, these correspond to caffeine. And as well in figure 7. And the hit quality index was relatively high for these, Product 1 was 80%. Product 2, which is figure number 8, gave a higher one to this, and it was like 96%. But products 2 to 6 gave no match against the library, which shows the presence of additional impurities in this. So what happened is, to source these products and to see what it's looking at, principal component analysis was carried offline to the spectra of the 6 products. And here we can see in figure number 9, in different colors, the 6 products relatively. And the orange and red are one product. The cyan and green one are two other products we can see, and then the purple and dark blue. What's interesting is the products that are grouped together show that they could be from the same source. So the orange and red can be from the same source, and the cyan and the green one. When we looked at the loading of the PCA, which is in figure number 10, we can see the peaks corresponding to caffeine. The one around 1500, the twin peak, is still there. And this was present in the cyan and green products, which we saw earlier in the figures 7 and 8, the spectrum of it, and gave high match value to caffeine. And this is interesting, the clustering of these products. It can help as well to track the source of these products, not just through looking at the overall signature, because there's a lot of mixture of substances that are scattering and might be characteristic to where it's made in this. And that was really important, because in one of the products we were about to find things scratched from impurities, scratched from synthesis making it. So this is good, because if we measure the sample by classical technique, once you dissolve the sample and filter it, all these impurities are gone. But this is the good thing about Raman. When they're measured by Raman technique, you get the spectra corresponding to all these impurities, provided they are Raman active. So this is in detail the PCA loading, the one that we saw in the previous picture. So it's in figure 11A. And then it's compared to caffeine in figure 11C, 2-aminoindane in figure 11B, and lidocaine in figure D. And it shows a high similarity between A and C, between the loading of the PCA in caffeine, which showed that it was the major impurity present in most of these products, at least the green and the blue one, which were overlapping, and they had high correspondence in the loading. And this actually was confirmed by reference analysis. So I would like to conclude this, that handheld Raman spectroscopy was really good for identification of illegal online products, which were counterfeit medicines, and as well tackling the impurities in novel psychoactive substances or legal highs. And both of these were a public health threat to the society. The internet played a major role in the spread of these drugs, as we can see the threats from online pharmacies and the United Nations report. The handheld Raman instrument, the inbuilt algorithms they offer, is really rapid tools for identifying the medicines. And we can see 2 different algorithms that were complementary for whatever matrix we're measuring, the different medicines or legal or drugs. Moreover, the offline analysis of MPS products with handheld Raman spectroscopy, it allowed the prediction of the major impurities, just like we found the caffeine, which was present in most. And it helped in tracking the source of manufacture of these products with the help of principal component analysis. And finally, I would like to thank BW Tech for the handheld NanoRam instrument, the BBC Television for supplying some of the internet samples, and the European Pharmaceutical Review for inviting me to speak. And thank you a lot for listening.
Thank you very much, Sulaf.
Thank you.
Now, don't forget that if you do have any questions for Sulaf or Catherine, please do not hesitate to submit these as we go along using the questions button. So it brings us on to our second speaker today, who is Catherine Bakiv. Catherine is Director of Analytical Services and Support at B&W Tech. And Catherine will be discussing the impact of handheld Raman for meeting PICS requirements. And it's over to you, Catherine.
Thank you, Matt, and thank you everyone for joining us today. Indeed, today I'll be talking about the advantages of handheld Raman in the pharmaceutical industry and how it can help us to meet the PICS requirements. The pharmaceutical industry is interested in Raman, indeed, because it gives the possibility to rapidly and portably make measurements of samples. Sulaf gave us some great examples about how it can help to protect the public health in looking at narcotic substances and counterfeit drugs. But indeed, there are regulations that require pharmaceutical manufacturers to ensure the safety and identity of their products from the beginning of the receipt of the materials into their manufacturing sites. Now, Raman itself, the effect was discovered 85 years ago by Sir C.V. Raman, but it is only 10 years now since the first portable Raman instruments were made available, and the miniaturization of the instruments continues, making it easier to use these instruments for many new applications, such as rapid identification of materials. In the industry right now, there are trends that companies must meet regulatory requirements as set out by the PICS initiative, which is the Pharmaceutical Inspection Cooperation Scheme, as well as the GMPs and pharmacopoeias of their own nations. PICS is a cooperation of 43 member nations, with many countries still applying to join, and the idea of PICS is to try to unify the regulatory GMP and inspection across the globe. So there's been quite a trend with increasing product contamination incidents, as well as counterfeits, recalls, and facility shutdowns. So companies really are in a need of developing rapid and specific tools that allows them to check and verify the identity of the ingredients and products that they're doing in terms of incoming material inspection as well as real-time release of their products. So the implementation of 100% testing of raw materials is coming. So indeed, as I stated, PICS is an international convention between countries and pharmaceutical inspection authorities, which provides an active and constructive cooperation in the fields of the GMP with the mission of unifying the standards globally. It currently has 4 partners as well as 43 member nations, with applications being received from many countries on a regular basis. In this map, we see then all the member nations as well as the partnering groups that are members of PICS. So PICS GMP requirements are actually based on the European Medicines Agency's and the FDA's GMP guidelines. And within those guidelines, it is stated that there should be appropriate measures in place to ensure the identity of the contents of the materials that are being used for manufacturing, as well as for sampling and identification of packaging materials. So this means that 100% container testing is moving to be the norm all across the globe as companies unify amongst the GMP requirements. Now at this time, I'd like to stop and ask a voting question, if we can bring that forward, please. Okay, thanks everybody for responding. So let's continue on now. Indeed, 100% raw material inspection is coming, but how can we rapidly perform 100% container identity testing? I'm sure many of you are familiar with technologies that enable one to do this, and indeed Raman spectroscopy is one such technology. It is a form of molecular spectroscopy, and it's based on the scattering of light by molecules. Because the molecular vibrations are specific to the composition of materials, a Raman spectrum serves as a fingerprint and is a very specific methodology that can be used for identification of materials that have molecules that are active and present a Raman signal. Here I show some examples of some Raman spectra of different compounds, with showing you which of the functional groups are giving rise to the peaks that are present in the Raman spectrum. Indeed, Raman spectroscopy can be used to measure many different types of materials. Many of the materials that you may encounter have a very strong Raman signal, such as active ingredients and alcohols and such. But indeed, there are materials as well that have a weak Raman signal and may fluoresce. So samples such as glass does not have much of a signal, nor does water, or samples that are very dark in color. That we can use to our advantage or disadvantage for the packaging materials, but indeed some materials such as black materials and metals and ionic salts have no Raman signal. So Raman is not the answer for measuring all materials, but it does cover a large proportion of organic and inorganic materials that one may encounter in the pharmaceutical industry. And due to the weak signal of water as well as glass and the ability to focus the Raman laser beyond the point of contact with the material, Raman is very good to enable one to measure through a broad class of packaging materials, as shown in this slide here. And here I'm showing the overlay of the Raman spectra of acetaminophen, where we've made the measurement of the acetaminophen with direct contact with the instrument and going through various layers of plastic packaging. And we can see that there is very little effect, very little signal actually, contribution from that plastic packaging, just reinforcing the utility of Raman as a rapid tool for measuring materials without having to prepare the materials or indeed remove them even from their packaging. Now, when we're doing Raman methodologies in terms of identification and verification, it's important that we have the proper samples in order to develop such methods. And Sulaf gave us some very good examples of how she collected samples for her studies. Having the right samples is quite similar for any type of laboratory methodology that you would want to be developing. But it's important in developing methods that you include the variability of the samples that you may be needing to verify in the future. And using a method on the NanoRam, allowing you to collect 20 scans, enables you to build up a robust method where you can consider the variability of material suppliers, different ages of products, different samplings and packaging, and indeed just the natural variability of products that may exist. And indeed, these guidelines for method development are those that are developed in the EMA and referred to in terms of near-infrared spectroscopy, which are quite relevant and applicable here for Raman spectroscopy as well. Then when we come to making our measurements themselves, we need to define the measurement parameters. And here we show that you can define those measurement parameters in terms of how much power you would use on the laser. And indeed, you have different sampling accessories that you can use with the NanoRam to be able to use the most appropriate accessory appropriate to the sample form to get the best signal, the best spectrum available for the measurements. There are different tools on the NanoRam for material identification and verification, as Sulaf mentioned. So when we're using the identification mode, what we're using is using a defined method with a p-value with a significance level set to 0.05 or a 95% confidence that a material is what we've measured it to be against the 20 scans in a method. So we use this to verify material identification. Indeed, we also have the possibility to develop and use purchased libraries to identify unknown materials using the hit quality index, that correlation of an unknown material to a spectrum that is in a library. One thing that is very important, regardless of which mode one is using identification or investigation, is that one gets very clear results and indeed also has the ability to view those results both on the instrument and to manage the data separately, which I'll talk about in terms of data management through a PC. Regardless of the methodology that one's using, it's critical to qualify the instrumentation to begin with, and the methodology that one's using as well. And here I show an excerpt from the USP 1058, which is showing that there are different aspects of qualification that are important for instrumentation. And all of these things can be accommodated, of course, with Raman spectroscopy. One important aspect, indeed, is verifying the performance of the instrument on a regular basis in terms of performance qualification, PQ. And on the NaNoRAM, indeed, one has this ability to run a validation sample. Typically, we use polystyrene, which is used, and one can get a pass-fail result. And again, in keeping in the importance of having a qualified instrument, if the instrument doesn't pass this, the user is then locked out because the instrument is not at the proper quality to enable one to collect data for methodologies. Now I'd like to pause again to present another poll question, if we may. Okay, thank you all for taking the time to answer our poll question. Now I'd like to show an example of making some measurements with the NaNoRAM. Indeed, we have the mode of developing methods where one scans 20 spectra of a material that one wants to verify the identity of. And with this, we use multivariate analysis to define then what the acceptance criteria is, the 95% confidence for materials. So our p-value then would be such that samples that have a p-value of greater than 0.05 would fall into the multivariate space of the material. Samples with p-value less than that, indeed, do not represent a member of that population and would fail the method. So once that method is developed, it is very straightforward to do the sample analysis, and this can be done by any level user who can then access the identification mode, choose the method that they need, and get a rapid pass-fail result. Now, one of the advantages of having this different methodology, the multivariate methodology, in addition to an HQI, is illustrated here where I have an example of some chemically similar materials. Here I have potassium carbonate and potassium carbonate sesquihydrate. And the Raman spectra of these, where we see that the predominant carbonate peak dominates the spectra, and the spectra are very similar, except for some additional peaks related to the hydrate that's here in the sesquihydrate. If we use the HQI library searching correlation methodology, we see that in all scenarios, one would get HQI values of greater than 95, so a correlation of greater than 95 of each one of these materials against the other. So in this case, the possibility of ambiguous or incorrect results is possible because of this very high correlation that one could get. In this case, turning to developing a multivariate method using our p-value methodology, we can see very clearly that when we test samples of potassium carbonate against the potassium carbonate method, we get p-values that are consistent and very much higher than the threshold value of 0.05, whereas if we test it against the potassium carbonate sesquihydrate method, we do get failing results with p-values significantly less than 0.05. So the ability to unambiguously and properly identify materials using the multivariate p-value methodology opens the possibility for very robust and accurate methodologies with the handheld Raman spectroscopy. Now again, regardless of the technology one's using, it's quite important to validate the methods that are used. And in developing methods, it's important to validate them in terms of testing them to ensure that the proper materials pass and that the incorrect materials fail. So this method validation must include challenge samples to ensure ongoing validity of the methodology, as well as to just make sure that the sample is truly specific and robust. Indeed, the European Pharmacopoeia chapter on Raman spectroscopy recommends that the selectivity of qualitative methods should be challenged on an ongoing basis to ensure validity of methods. The International Conference on Harmonization also provides guidelines on validation of analytical methods, and here I summarize basically stating that all analytical methods should be validated, and the suitability of the testing methods used should be verified under actual conditions of use and documented. With this in mind, we have developed protocols and guidelines to help users to develop and validate the methods that they would develop with their handheld Raman spectrometers. And indeed, when we're looking at qualitative or identification methods, again, I'm referring to the guidance documents of the ICH. And here the important test for validating an identification method is to show the specificity of that, which, as I previously stated, can be done by challenging a method with known good samples and known bad samples, which could include name analogs. So at this time, I'd like to pause again to open the possibility for you to vote on a question. So if we can ask you to please take a vote. Okay, thank you everyone for taking the time to answer that question. Now I'd like to move along and just talk a little bit more about some of the important aspects, methodologies, and some of that includes the data management and reporting. When you're dealing with any device, of course, one needs to have secure data and a secure data management system. With the NanoRam, what we use is indeed a PC-based software, which we call NanoRam Identification, ID, where one can synchronize data back to a server, and from this generate reports, look at the audit trail, look at all the reports, and keep track of all of the results that have been generated, as well as the methods and libraries that are resident on the handheld device, but those data are now stored on a server system. So again, very important aspects for methodologies. The reports that one can generate also include all of the very important information about the measurement, including when the instrument was last qualified, when the PQ was last done, as well as a date and time stamp of when the measurement was done and when the report itself was generated. And one can also generate summary reports for many measurements that have been done together. So with that, I'd like to thank you all for your attention and hope that we've been able to show that Raman is a promising tool to increase your operational capabilities and reduce your costs while providing rapid material identification possibilities. So thank you very much for your attention, and we'll be happy to address some questions at this point.
Yeah, excellent. Thank you very much, Catherine. So yes, as Catherine mentions, we've had a number of great questions come in. So hopefully Catherine and Sulaf can answer some of those for you. So for the question and answer session, I will be handing you over to Stephanie Nave from B&W Tech. And yeah, over to you, Stephanie.
Thank you, Matt. Okay, so just in order here, one of the first questions directed at Catherine is, how is it that Raman can be made so small?
Some of the advances in the technology that enable Raman to be made so small are indeed the smaller detectors that are available and the ability to cool these very readily, advances in the lasers, and in all of the electronics, including the electronics such as is being used in many of our other handheld devices we have in our lives. So with the advancement in electronics in terms of just the battery power and the screens and such, as coupled with the detectors and the lasers that are used in the Raman, enable this small packaging to continue to be even made smaller.
Thank you, Catherine. Another question, when you perform the PV using the polystyrene sample, is it possible to obtain the actual peak positions of the polystyrene bands? If not, how can you show you are meeting EP requirements?
Thanks for that question. When you're doing performance verification on the NanoRam, what we do actually is we do compare the peak positions as well as using a hit quality index, an HQI, to compare the polystyrene PQ with the factory-created polystyrene spectrum. If one wants to go exactly against the peaks and take those data out. One can do that, but we are doing that and reporting that, and that is what makes up our passing of the PV testing.
Thank you, Catherine. Another question, how would you compare this technique with portable NIR spectroscopy? And actually that one's directed at Sulaf.
Okay, so actually the right thing is not to compare them. They both serve as complementary. But the advantage of Raman is that when you are looking for specific drugs in the field, like using drugs of abuse, it can be selective for the drug, and the libraries can be built up to subtract the excipients, the additives from them, or inactive ingredients in them. But you cannot compare Raman versus NIR. I'd rather use them as complementary because each technique looks at different types of samples.
Thank you, Sulaf. Next question, can I export results to a LIMS system? In particular, can I set up automatic reporting with your database?
The NanoRAM software does support the ability to automatically export data when you synchronize with the computer into a format that's compatible with LIMS and can go directly into a LIMS database. So indeed, there is compatibility, and it is quite easy to set up for automatic updates and transfer of the data on synchronization to a PC into a LIMS system.
Okay, great. Thank you. Has there been any information on when 100% container testing will come into play?
That's an interesting question because indeed in Europe in the 1980s already the European Medicines Agency had called for this, and that was when a lot of spectroscopic tools were introduced to try to address this. It really is up to each member nation's regulatory authorities on when they will define and how they will define this, but we're seeing very much activity globally in moving forward to do this. Some of the issues that arise with this are, of course, the absolutely large volume of testing that this is putting forward. But there are also many nations that to this point have done very little testing of their incoming raw materials. So for them, even if it's not 100% container testing that they will adopt at this point, the fact that they will adopt testing schemes of some sort of sampling of incoming materials will help to ensure the safety of the medicine supply globally.
Okay, great. Thank you, Catherine. Someone else had asked, I have a light-sensitive sample. How would I measure that with Raman?
As mentioned, Raman is not very good for measuring dark samples. Indeed, you are using a laser to excite the molecules to create a Raman spectrum. So if a sample is light sensitive, one may want to turn down and use a very low laser power to enable to measure that. But there are some samples, such as very dark materials, for which Raman is not suitable.
Another question here, how would you compare Raman with handheld FTIR?
Again, as Sulaf said, I would look at these as complementary tools. Indeed, when we learn spectroscopy in school, they are taught as such because there are different molecules that are Raman active and FTIR active. One of the limitations of FTIR is actually that it is not so good at measuring through packaging. So Raman has the flexibility then of being able to measure through packaging, and really that packaging can contribute a little bit, but I think I showed you an example wherein you can adjust the focus of the laser such that you're going through a packaging that could be a few millimeters thick. Therefore, you're not actually getting the signal of that packaging, but of what's in that package. FTIR doesn't have that capability. It's important to have direct contact, and FTIR enables one to have intimate contact with samples in order to measure a good quality spectrum.
Yeah, as well, I would like to add something. With handheld FTIR, it's very important to crush the samples. So for measuring authenticating pharmaceutical tablets, not only you have to have the tablet as it is without the packaging, you need to crush it as well. And this is where Raman is better. It's non-destructive in this case, if you don't want to destroy the samples.
Another question, is it possible to transfer a reference spectrum of a new substance acquired on one instrument to another instrument, or share it with many other users having the same kind of Raman analytical device?
Indeed, within the NanoRam and the data management software, one does have the ability to export data, and it can be exported in different formats that are compatible with other softwares for further analysis. And I believe that is part of the analysis that Suloff herself did in showing us the different signals within the drug substances she was looking at. But yes, one can transfer reference data and transfer libraries from one device to another. It is best to do that of devices of the same technologies, of course. So one thing is the proprietary nature of the data formats that companies use, but the other thing is that there can be slight differences in the spectra collected on one instrument to another. So though it can be done technologically, I would not say that it's necessarily advisable, because indeed measuring the spectrum of a new sample on the same device may be quicker and may give more accurate and precise results.
Okay, can you indicate the cost, price of these handheld devices?
These types of devices range in price from about $15,000 to $50,000, depending on who's supplying them and what type of accessories and such, and the size of them that one using. So really one needs to look at exactly what your needs are, and then look to get the exact price for what you need for your solution.
Okay, someone had asked, how long does it take to measure a Raman spectrum?
Okay, it depends on the sample you have. It's really quick, within seconds, but if something fluoresces, it might take a little bit longer time, but normally it's really fast, less than a minute.
Okay, great. Could you talk about some Raman applications other than pharmaceuticals, such as the petroleum industry?
Raman spectroscopy has been used widely in many different application areas. Indeed, in the petroleum industry, it's been used for measuring the different oxygenates in fuels. Not so much with the handheld Ramans yet, but if one goes into the history of Raman, going back for 85 years, one can clearly see that it's been used in so many different application areas that we could actually easily do quite a longer session on just talking about different applications. But within our portable and handheld devices, it is used in the plastics industry, in biomedical diagnostics, for instance, in identification of gemstones, in looking at art and archaeological artifacts and identification of the different pigments that are used in those, and the list goes on and on.
Okay. Does handheld Raman spectroscopy require a minimum sample weight or size to give correct measurements?
That's an interesting question, and I would say it's not about the weight and size, but really about the components that are in the instrument. So I guess that means that the instrument size, or is it the sample size? Because the sample size needs to be such that you are actually covering and measuring across your whole beam that you're looking at. So it doesn't need to be very large, but I can't give you just off the top of my head a specific number on what that would be.
Okay, great. I think those are about all the questions that we have. So back to you, Matt.
Okay, fantastic. Well, thank you very much, Stephanie, for handling all the questions there that come in. I saw that we do have quite a lot of questions. So for the ones that we didn't answer, we'll try and get these out via email to all those registered. We can continue the discussion after the webinar. So yes, as mentioned, unfortunately that's all we have time for today. I'd just like to say a big thank you to B&W Tech for their support. Obviously to all of our speakers, Catherine and Sulaf, and Stephanie for handling the Q&A session. And of course, thank you all for taking the time to come and listen to our interactive webinar this afternoon. So again, just to mention, this webinar will shortly be available on demand. So if you did find it interesting, you can always share it with your colleagues. And finally, this has been me, Matt Cheeseman from the European Pharmaceutical Review. Thank you very much again for listening, and I hope to speak to you all again very shortly at one of our forthcoming webinars. Thank you.